Good afternoon, everyone, and thank you for joining us today. My name is Carola Schönlieb. I'm Professor of Applied Mathematics at the University of Cambridge and Turing Fellow. At Cambridge, I'm leading several data science initiatives, among them my own research group, the Cambridge Image Analysis Group. I'm the director of the Cantor Capital Institute for the Mathematics of Information and director of the EPSRC Center for Mathematics of Information in Healthcare. Today, I'm going to wear my Turing Fellow hat, and it's my pleasure to be chairing today's Turing lecture. The Turing Lectures is the Institute's flagship lecture series, which have been running since 2016 and virtually since April 2020. They are free for all to attend, and we aim to make them as open and accessible as possible. Before we get going, a few housekeeping notes. Today's event will be subtitled. If you can't see subtitles and you would like to, hit the CC button on the bottom toolbar of your screen. And if you'd like to view subtitles in the new screen, follow the link in the chat. If you are on Twitter, you can discuss and feedback on the lecture using the hashtag #TuringLecture. And don't forget to tag us at TuringInst. You can submit questions via the Zoom Q&A function throughout the lecture. And if you'd like the opportunity to join the panel and ask your question live, please let us know by just typing live at the end of your question. We are expecting a lot of questions this afternoon and will not be able to get through as many as we'd like. So apologies in advance if we did not get around to your question. But we really encourage you to ask questions and possibly also upvote other audience questions that are similar to your own to ensure all the important topics are covered. We will be recording this lecture, and it will be hosted on YouTube after the event. So if you need to log off early for any reason, you will be able to catch up later. Finally, the chat is open, but please be reminded of our events code of conduct and keep all messages friendly and professional. And a link to that is going in the chat now for further information. So now, without further ado, it's my great pleasure to introduce our esteemed lecturer, Professor René Vidal. Before I hand over the word to you, René, let me just say a few words about you, your research expertise, and your several research leadership activities and contributions that you have made over the years. René Vidal is the Herschel Seder Professor of Biomedical Engineering and the inaugural director of the Mathematical Institute for Data Science at Johns Hopkins University. He has secondary appointments in computer science, electrical and computer engineering, and mechanical engineering. He's also a faculty member in the Center for Imaging and Science, the Institute for Computational Medicine, and the Laboratory for Computational Sensing and Robotics. Last year, Professor Vidal was elected to the American Institute for Medical and Biological Engineering's College of Fellows, which represents the most distinguished and accomplished leaders in the fields of medical and biological engineering, based on his outstanding contributions to medical image analysis and medical robotics. To drill a bit deeper here, René's research focuses on the development of theory and algorithms for the analysis of complex, high-dimensional datasets such as images, videos, time series, and biomedical data. His lab has pioneered the development of methods for dimensionality reduction and clustering, such as generalized principal component analysis and sparse subspace clustering, and their applications to face recognition, object recognition, motion segmentation, and action recognition. His lab creates new technologies for a variety of biomedical applications, including detection, classification, and tracking of blood cells in holographic images, classification of embryonic cardiomyocytes and optical images, and assessment of surgical skills in surgical videos. His current major research focus is understanding the mathematical foundations of deep learning and in its applications in computer vision and biomedical data science. And now, without further ado, it is my pleasure to introduce Professor René Vidal to the Turing Lecture stage for his talk on engineering the future of medicine. René, over to you.
Thank you very much, Carola. This is really a great pleasure to be here. I appreciate very much the invitation, and I'm very honored to be giving this Turing lecture. Before I start, just wanted to make sure that you can see the slides in full screen and everything's working.
Yes, we can see them full screen.
Great. Let me get started then. Imagine what medicine might be, say, 30 years from today. I imagine, for example, what might be May 19, 2045, when it'll be the 150th birthday of Johns Hopkins. How would maybe our medical appointments look like? Perhaps we're just gonna go to the doctor and we're gonna have something like an Alexa assistant that is going to be right next to the doctor. They're going to be listening to the conversation. The doctor is going to be asking us questions, and depending on the questions and our answers, the doctor will ask us new questions. Perhaps Alexa will be in parallel looking for similar patients in a very large database, looking for what treatments were given, what worked, what didn't, perhaps suggesting to the doctor a question that he forgot to ask, and maybe suggesting a few tests that need to be done, maybe looking also at my medical history, and being able to ultimately suggest potential treatments in collaboration with the doctor. Moreover, it may even be able to book our next appointment so that we don't have to call again. While this may seem like something really far in the future, and indeed we're very far from getting to that vision, we got a little closer this year with COVID, and I personally had my first online appointments that I could do and just do the appointments online and then have a telehealth visit. But we are very far. We are very far. Nonetheless, there is a lot of promise that this might be closer than we had anticipated. Why? First of all, I think there's been a great trend in the market regarding the dramatic growth of the use of AI tools in healthcare. And this is driven by a variety of companies. There are many more here, but I'm mentioning only two, Google and IBM. And if we look at the statistics of what was predicted in terms of the growth of the AI market, the prediction had been that there was going to be an 11-fold increase from 600 million to 6.6 billion in the US. And interestingly, so these predictions were from a while ago. If we now look at what's happened, this 6.6 billion was actually achieved in 2020, 1 year earlier. And the predictions are that, again, in the next 7 years, there will be, again, a growth of the order of 41% in terms of the growth of AI techniques, the market in AI in healthcare. And this is only in the US. So what is the type of AI and what AI can do for medicine today, and how far are we from what we envision that might be possible in the next 30 years? So to AI, it's really significantly simpler than what I said. Today, AI, by and large, is we need to collect a lot of data. This data might be of very different varied sources. It may be time series like EEG or ECG. It may be a rich variety of different imaging modalities at all different scales, from the microscopic, like calcium imaging, to the macroscopic, like diffusion MRI. It may also be, as illustrated in this video, data that comes from a surgical robot, where you might be looking at the movements and the trajectories or videos of what the surgery looks like. Or it may really be the electronic health records, which is what I was trying to do. And that data needs to be labeled and annotated with whatever we might want to predict. So we might want to annotate it with the phenotype of a cell, if it is an image, or maybe in the case of the surgeon, we might want to annotate it with what's the level of skill of that surgeon at doing that surgery. Or in the case of electronic health records, we might have different diagnosis, prognosis tests that were done, and so on and so forth. So being able to take all of this data and make predictions that can help diagnosis is incredibly challenging. And there are many challenges that are associated with this problem. One of them, I've already mentioned it briefly, is the great variability of and diversity of different kinds of data that operates, different sensors operate in dramatically different principles, and they are looking at dramatic different scales in the human body. From the microscale, where you might have scattering properties of molecules, all the way to the full body via PET, CT, MRI, or very different kind of data, as it might be electronic health records that just have, say, the doctor notes during your visit. Another challenge, and this is a little bit conflicting, I've written here big and small data. What I really mean is big data and small annotations. So there is no doubt the data is big and very complex. If you think about the number of procedures that are done every year that involve, say, one medical image, the growth of those procedures of 20 to 40% per year, the number of medical images that are stored in the US alone, and the fact that it is predicted by NTNT that about a third of the global storage is just going to be medical images. So these images are very large, even much larger than what is typically used in computer vision. Images in pathology might be, say, 4,000 by 4,000, and similarly, brain MRIs are three-dimensional, and there are even some images that are four-dimensional if you have time. But the challenge is that while that data is collected, it is not shared. And the fact is that it's a lot of data, but from a single patient is very sparse, because it's not the case that we take an MRI of a patient every month or anything like that. It's, every few years there might be one of those. And so when it comes to tracking those anatomical changes for a single patient, we might have only one image or two or three. And secondly, the ability to annotate those data sets is very costly and very time consuming. So for regular images, such as those that are available in Facebook, we can use humans to annotate them. We can upload them in Mechanical Turk and pay, say, 1 cent per image to get, say, a bounding box around every object that is present. But for medical images, generally we need experts. We need a pathologist, we need a clinician, we need a radiologist that is really looking at those images and making decisions. And sometimes finding something might be like finding a needle in a haystack. In contrast, pretty much all of the machine learning methods that are being developed today are data hungry. We need a lot of data, and we need a lot of annotations. And the more annotations we have, the better those methods operate. Therefore, there is here a big challenge of how are we going to translate computer vision and machine learning technology into the medical domain when, by and large, the expectation is that we will continue to live in this world where the data will continue to grow in complexity and size, but the amount of data that actually can be labeled will not grow as dramatically. Last but not least, it's the question that most machine learning methods today are not interpret. They make predictions like this is a tumor, this is not a tumor, and the issue is that when they work, and when they fail, but we don't know why they work when they do, nor do we know why they fail when they fail. And if you like to translate these methods into clinical practice, you would like medical doctors to understand those predictions and trust them. And therefore, we need to figure out ways of making those predictions more interpretable. Of course, It's also very important that these predictions have some guarantees of accuracy and that they need to be safe for the patient, and they need to make decisions that are also fair, that don't discriminate across different populations. And these are all really growing areas in AI and machine learning and computer vision, which is how do we make those methods more interpretable, safe, and fair. And those are areas that many of us are actively working on. So moving forward, I think there will be a strong need for building platforms that allow one to share data in a way that is much more easy than today, that allows to make the annotation of data significantly easier than it's possible today, and a feedback loop of interaction between clinicians and data scientists where data can be collected and cleaned and annotated much more easily for the data science to work, and data scientists can produce results that can be quickly interpreted by clinicians to provide feedback as to the usefulness of those methods. On the data science side, we need to develop methods that require less supervision that is possible today, meaning they need to operate with significantly less labels, and they need to be more interpretable and with guarantees of performance. So with that introduction in mind, I wanted to get of where we are today, or at least the work that many of members of my team have been doing over the last 10 years in various areas of biomedical data science. So I'll talk first about work that we've done at the cellular level, where we've been looking at a variety of different problems. I will speak about the first project on the top left, where we've developed a new blood test. And this new blood test is developed using techniques from reconstruction, classification, and detection in holography. I will speak briefly about this project on the right that is more machine learning and neuroscience. How do we take complex data and are able to detect where neurons are and their spiking activity? I won't have the time to mention, but we've also been working on the question of classification of stem cells from optical images and being able to determine different phenotypes from embryonic stem cells and try to predict what kinds of cells they will grow into once they mature. I also won't be able to talk about the work that we've done in brain MRI, but we've done a lot of work in diffusion MRI, particularly questions such as reconstruction and compressed sensing in that space. And I'll conclude my talk really with the work we've done in surgery, in particular using videos as well as motion data from the Da Vinci robot to be able to assess the level of skill of surgeons. And I'll finish, I think, with just one slide about what we're currently doing, trying to help children with motor disabilities as well as autism by using computer vision tools to estimate their movements and using that to help diagnosis. All right, so let me begin with the first project, which I've called here Machine Learning in Hematology. And first, in full disclosure, this work was supported by a company called My Diagnostics that is actually developing the blood test that I'm going to be talking about. I imagine many of you have this device at home, and this is probably the device that is used the most for diagnosis today. We just take our temperature, and we know, let's say you have children at home, that as soon as the temperature is 100 degrees, then you've got to take that child to the doctor. In contrast, this is probably maybe the second most test that is used for diagnosing a variety of diseases, and this is the blood test. And we don't have a hematology analyzer at home. The process by which we get that test today typically takes several days, even though the test itself might just be a matter of 30 minutes at most. In particular, we usually go to the doctor's office. They might say, okay, you need a blood test to be diagnosed. Then you might need to go take the test either the same day or more likely than not the next day. And then it might take another day to get the results. And then you need to schedule yet another appointment in order to get the results. And for those of you who might not know, the complete blood count is one of many blood tests, maybe the simplest and the more broadly used, that produces concentrations of different quantities, including the white blood cells, red blood cells, platelets, hematocrit, and whatnot. So the goal of this project, and this is a collaboration between this company called My Diagnostics, IMEC, which is a research center in Belgium, and Johns Hopkins University, is to develop a new blood test that might operate just in a matter of minutes, and therefore could be done at the doctor's office or perhaps even at home. The idea is to take a small drop of blood from your finger and then pass that through a microfluidic device that will then you will insert that microfluidic device into a chamber where it can be imaged. And for various reasons that I'll explain in a moment, we're using a technique for imaging called holography that ultimately at this level, you can just imagine it produces a video with thousands or hundreds of thousands of cells that are moving through the microfluidic device. And then from a machine learning or AI or computer vision perspective, the goal is to take these holograms and be able to produce automatically the blood count. So why holography? Why not use sort of the standard that is used today? That is, you put a blood sample under a microscope and then you're able to look at the cells. Well, the main challenge really is that in microscopic imaging, you can see the cells, and you can see, for example, here, these are red blood cells, and you might appreciate that the shape looks a little bit like a donut. And the issue, though, is that you can only see so many cells at a time. And if your goal is to count and produce concentrations, you need to get a very large number of cells so that your estimates of concentration are more reliable. And so there is this trade-off in which you can use lens-free imaging that is illustrated on the right. That lens-free imaging, that's a very small portion of a real image for you to compare. And therefore you can image a very large number of cells simultaneously. But as you can appreciate, the resolution of it is very low. And therefore your ability to really appreciate what the cells are and how to detect them is very hard. So that's exactly what happens. On the left, you can get very high resolution, and therefore classification might be easier, but that will lead to what is done today. You need microscope, you need lenses, therefore the apparatus is going to be bulky and expensive. On the right, if you use holography, you are going to have low resolution, but then the size is going to be very compact. You don't have any lenses, so you can image a very large field of view, and therefore it's lower cost, and you can build a device that might just be the size of a USB drive. So just to show a little bit more detail as to what these cells look like under the microscope, these are examples of white blood cells, and you can see that there are many kinds, but these are sort of the three main phenotypes, and you can begin to appreciate some differences, like granulocytes have many nuclei, while monocytes have only one. Lymphocytes are a little bit smaller. But just for you to contrast the resolution, this is how they look from lens-free imaging. So here you can see all the details. Here it's really very pixelated. And so a first challenge we needed to face was the question of, we really need to figure out much better reconstruction techniques that would allow us to take very large images. Again, what I'm showing on the far right there is a very small fraction of a very large image, and to produce reconstructions that are significantly better than what is shown here. And so just for reference, what you see on the right, the black regions are the ones that correspond to the cells, and everything else is a bunch of interference patterns that arise due to the acquisition protocols in holography. So this is what we were able to do. So by exploiting what we knew about the kinds of images, namely the word sparsity, So what we want, or what we knew, is that we were going to have very large images, but most of it really should be background, and only a small fraction of the image should be occupied by cells. And therefore, by using sparse reconstruction techniques and enforcing sparsity, we were able to get the reconstructions that you can see on the right. These techniques should be somewhat similar to those of you who are familiar with the field of compressed sensing, but with a number of modifications. One of them, which is illustrated here on the top left in this equation, so H here is a hologram, so this, you can think of it as a 2D image if you like. But this, what we want to reconstruct is something that is 3D and in the complex domain. So this capital X is really a 3D volume, but it's also complex. And these filters are complex valued filters. And so this phase W is the phase, and that is unknown and also needs to be recovered. So that makes the problem non-convex. You also have filters, so it's a sort of convolutional sparse decoding problem in the complex domain, with constraints that pertain that the phase needs to be unit norm, and you also need to enforce sparsity. So anyhow, we were able to develop efficient techniques for solving these problems in the complex domain, and that are scalable, and that produce the results that you can see here. So again, for comparison, you can hopefully you can appreciate the significant improvement in terms of reconstruction. And you can even begin on the far right. These are red blood cells. So you can already begin. It's not perfect, but you can begin to appreciate the donut shape that is present on red blood cells. So having worked on and significantly improved the reconstruction, the next thing we needed to do was to develop algorithms that would allow us to determine, is this a red blood cell? Is this a white blood cell? And if it is a white blood cell, what kind of white blood cell it is? In this case, we have 3 different phenotypes: granulocytes, lymphocytes, and monocytes. And even though the quality of the reconstruction had improved dramatically, I hope you appreciate that it's still between granulocytes and monocytes. It's very hard to tell. In contrast, you can do that very easily when you're looking at microscopic images. But beyond that, there were many challenges that I'm illustrating here. So that's how one cell looks like. But as we continue to zoom out, I want you to appreciate now the scale at which we needed to operate. And this is just one image of an entire video of cells in motion. And so we've got a very large field of view. These images are something like 1,000 by 4,000 pixels. If you are familiar with object detection in computer vision, right? We're used to detecting 1, 10, maybe 100 objects in an image. Typically, those objects need to be sufficiently large. Let's say at least 50 pixels by 50 pixels in order to detect them. Here we are in a situation where we have probably 10,000, maybe 50,000 objects in one image. The objects are not very large at all. You appreciated in the previous slides that there might be 5 by 5 pixels. Some platelets here might be 3 by 3. Maybe the white blood cells might be 20 by 20. That's the size at which we're operating. So the objects are very small. And last but not least, and this is what I highlighted at the bottom left, by and large, almost all state-of-the-art object detection technology in computer vision requires you to label each and every object in the image with a bounding box. Here, we could not afford that. We could not have a pathologist mark 100,000 cells for each frame of the video and tell us, is this a platelet, or is this a granulocyte, or is this a red blood cell? Moreover, that was not even possible, even if we had an army of pathologists, and even if they charge us 1 cent per annotation, as we could do in Mechanical Turk. Pathologists are used to looking at these images of very high quality, and their ability to annotate this quality was nearly impossible for them, particularly telling whether you had a platelet or not. and telling between granulocytes and lymphocytes at this resolution was very, very hard. So therefore, we need to begin thinking what are alternative approaches to state-of-the-art object detection in computer vision that would allow us to solve this problem. And trivially, because we had improved the reconstruction so much, we thought, okay, well, we can just do detection by thresholding the input image, and then we will be able to build templates, or in this particular case, we could even have filters or build a convolutional network that would allow us to predict the labels. Actually, let's suppose that we could annotate. We could have the pathologists give us labels for some fraction of images. Then this is what we could do. Just do detection, pass them through our convolutional neural network, and predict the class names. As it turns out, this works, but the accuracy is not high enough. And I'm going to be showing the numbers in a few minutes. So what did we do instead? What we did instead was, again, to exploit knowledge and exploit biology and exploit knowledge about the problem that you're trying to solve and put that together with the machine learning techniques in order to be able to solve the problem. So in particular, we began by building a probabilistic generative model of how images of these cells would look like. For example, you could say, oh, first of all, I'm going to draw a random variable that is just the number of cells in that image. And that number of cells probably depends on the size of the image and probably depends on the concentration. And in fact, that's probably what I'm trying to estimate at the end of the day. Now, given the number of cells, I can draw what is the class for each one of them. Let's say it's a red blood cell versus a white blood cell. I can draw the location of the cell, and I can also draw, we modeled it with an additional extra variable for the detection strength, which you can think of as a number between 0 and 1, where 1 means that the cell is clearly visible. And in that reconstruction, this means that the reconstruction value is very high in the 0 to 1 range versus very weak detections that are alpha is closer to 0. Then, because we didn't have a lot of annotations, we were trying to do an approach that was based on building templates. And the templates, akin to those of you who are familiar with dictionary learning, these would be atoms in a dictionary that we wanted to learn. And the idea was that there would be different templates for different kinds of cells. And again, here, this is exploiting something that is different in computer vision, where in computer vision, typically you want to detect very few objects, as I said, by few, let's say 50, but there is large variability of the different instances of object classes. For example, if you're trying to detect a pedestrian on the street, then they might be dressed with different clothing, there might be different sizes and whatnot. So when it comes to these cells, they're a lot more repeatable. And therefore, the idea that I could have a template for each one of these cells makes sense. And therefore, I have this generative model for images now that goes from drawing a number of cells to drawing their class, their location, and drawing a appearance-like template for each one of them. Now, we put together this probabilistic model that has some parameters with the detection part for the images, but we still needed to learn a bunch of parameters for this probabilistic model, and we still have the challenge that we didn't have annotations for doing those. In fact, we needed to do this with the following annotations. Just in principle, the concentration of red blood cells that you can obtain by taking the same sample and putting it in a hematology analyzer, and you're going to get the concentration and classification. So in other words, we needed to do detection at the level of individual frames of the video, but we only had annotations at the level of the whole video, like the total concentration, one single number per video, as opposed to one per bounding box of each and every cell in the video. So we needed to work with biologists to be able to collect data that is even separate from the data that we're going to test, to be able to get some form of ground truth. And as it turns out, and I am not an expert on this by any means, so I learned this by working with biologists, there is a variety of techniques that can be done to take a mixed population of cells, and through a process known as purification, you can get cells that correspond to only a single class. And therefore, we could take, in this case, let's say white blood cells, and then purify them into monocytes or lymphocytes or granulocytes, so that we could then image videos that would correspond to only one class. And therefore, we would know that all cells in those images would correspond to the same class. Now, that's in theory, because this purification process is not perfect. So let's say it works 90% of the time. So that means that you might have 10% of the cells in that sample that are still from one of the other classes. And again, we don't have annotations at the level of individual cells. So you're just being told, okay, all of these cells are monocytes, but by the way, 10% of the labels are wrong, but we don't know which ones. So we still needed to work in this situation of, but that was much better nonetheless than having labels at the global level for a mixed population. The other challenge is that there is this sort of domain adaptation challenge, in the sense that we are going to train our system on this purified data, but we still need to test it on images of a mixed population. So we needed to figure out ways to do that. And so we added a lot of priors also on the expected proportions of these kinds of cells that we could get from a very large training set of more than 300,000 patients from Johns Hopkins. So let me conclude by just showing a few numbers of how well this works. So the different columns are different methods. The convolutional neural network method is the method that I mentioned at the beginning. Just run every image through a thresholding to get a detection, and then classify each detected cell. And the second one, because our method of reconstruction was really sort of a convolutional sparse coding style method, we thought we can just do convolutional sparse coding for classification as well. And the last one was, if you wish, adding this top-down probabilistic graphical model on top of convolutional sparse coding. There is two sets of numbers. So the top part of the table is samples from normal patients, and the bottom part is all kinds of patients, both normal as well as abnormal. And I think the main message that I hope you can appreciate is that by putting together biological knowledge and incorporating that in the form of a prior model, together with machine learning techniques, you're able to get improvements relative to the pure data-driven approach of just passing the images through a convolutional neural network. In particular, the error in classification of the three kinds of cells we were looking at, in this particular case is the prediction of their concentrations, went down from roughly anywhere between 12 and 30%, which is what the CNN did, to roughly around 4, 5, 6, maybe up to 7% error with the proposal. So in summary, I just wanted to say that this is, I think, a great opportunity to put together teams that involve both clinicians, biologists, as well as people that work in hardware in the imaging side, as well as machine learning and computer vision, to develop a new blood test that might be able to make predictions that are not as accurate as those that are made with a hematology analyzer, but that would provide much faster access by being able to do a rapid test that might be available just in a matter of minutes. And again, My Diagnostics is the company that is currently developing this technology into a product. Let me move next to a different topics that is machine learning and neuroscience. In particular here, the idea is how do we develop AI techniques that can be used by neuroscientists to make predictions? In particular, if you think about what is it that the kinds of data that many neuroscientists use, traditionally, they would be able to measure individual cells with a single electrode, and that would provide very high resolution information, particularly in time, for the spiking patterns, for example, of individual neurons. A second approach is to use functional MRI that allows you to image the whole brain activity, and that provides you significantly better spatial resolution in a sense, because you can now image simultaneously the activity of multiple cells. But the spatial resolution actually is not that good. Typically, there is an averaging that needs to be done over hundreds or thousands of neurons. And therefore, you get maybe very good temporal resolution, but very bad spatial resolution. So there is this intermediate solution of using electrode arrays where you can still image multiple neurons simultaneously. You don't have information for every individual cell, but you have still better spatial resolution than doing it with functional MRI. And in particular, this is an example of a technique known as fluorescence microscopy, where you use calcium, and so calcium binds to the cell, and the idea is that cells or neurons in this case are flashing whenever there is a spike. And I hope you can appreciate two things. Number one is that the images, again, are very, very large. So again, this can be several thousands by several thousand pixels. But second, and as before, the resolution of the images is very low. And so you can probably see in the zoomed version on the left that there are multiple neurons, and eventually there is a large neuron that flashes. There you see it on the bottom left. But there are a couple more, and what the neuroscientists need to do in order to do their analysis is, number one, to detect or segment or find the shapes of those neurons, as well as their activation patterns. And they need to do this in a situation where the resolution is very low and you have a lot of artifacts. So when Ben, who is the student that developed the approach for doing this, looked at the problem, he began by noticing that eventually what you need to do is sort of disentangle the spatial component that would tell you where the cells are located, because the cells are not moving, so they are static, versus the temporal component that is going to tell you the spiking pattern. And once you begin looking at that problem, you would say, oh, I can decompose the video. If there are 50 cells, then this is a superposition of 50 videos, one per cell. And moreover, looking at an individual cell, as shown here at the bottom right, there's just going to be a single image that describes that shape component. And the product of that single image with the temporal variability for that cell is going to give me the video for that individual cell. And then I'll add them up to produce the video for the 50 neurons at the same time. And so that's exactly what we did in terms of modeling. We begin with a video sequence here, Y. And what we want to do is to estimate a shape image that corresponds to an individual neuron, as well as the spiking pattern. And the observation is that both of them are sparse. Much of the shape image needs to be background, except for the regions that contain the cell. And much of the temporal activity needs to be at baseline. And only when there is a spike, we would like to have a signal. And therefore, the question is, can we enforce sparsity both in the shape of the image as well as the activity of the neuron? Now, it's a little bit more involved than that because you have to consider the dynamics of calcium. So it's not just a spike, but there is an exponential decay. So it's really like taking spikes, but convolving them with some dictionary that corresponds to the activities or the calcium dynamics And so modulo that, you can go back to classical linear algebra and matrix factorization techniques, really, where you say effectively that the video is a sum of rank 1 videos. In rank 1, because each one of them is sort of the product, as I already articulated, of the shape image and the temporal activity. So think about the shape image as a vector now, and think of the spiking activity as another vector. So that's exactly what UIVI transpose does. And D is going to be the convolutional operator that captures the calcium dynamics. And R here is going to be the total number of neurons. So you would say, oh, let me just do some sort of PCA and the rank is going to be the number of neurons. We did all of that and it fails miserably. And it fails miserably because the principal components do not capture individual cells, which is what we want. And so we came up with an approach where we exploit structure that is present on the problem. So we knew that we wanted the shapes of the shape images to be sparse, which we can capture with a L1 type of penalty. We also want the images to be compact, right? I might have 5 non-zero pixels, but I don't want them to be all over the place. I want them to be concentrated to form that cell. So that can be captured with another norm on U that is known as total variation. And we also want the spiking patterns to be sparse in time now. And so with that in mind, we posed this denoising or problem as a matrix factorization problem. It's actually convolutional, but for simplicity, let's think about matrix factorization while enforcing a variety of priors on the factors. And as you can see, on the far left, you've got the raw data. That's the one that was very noisy and very hard to detect when things are happening. If you add only sparsity, you get what's in the middle. You can also add low rankness because you want the smallest number of cells, and you can add total variation. And I think the main message that I'm trying to deliver is appreciate the quality of the reconstruction that can be obtained on the far right, where you can now see much more easily when this, there is a spike, where the cells are located, and you can also determine the shape. While all of this work came up from a problem in neuroscience, and I'm not mentioning here, it led to a lot of work in the pure math side, where for those of you who are mathematicians, you would appreciate that this is a non-convex problem. It's also non-smooth. And you might wonder, well, can I have guarantees of global optimality? Can I find the optimal rank? And so we did a lot of papers really on non-convex optimization, where we now have guarantees of global optimality for this type of problems. And in fact, all of that theory was even extended to deep networks, because we were able to show that these low-rank matrices, low-rank tensors extend with nonlinearities to the analysis of certain classes of deep networks. All right, let me move now to the third and final topic, or at least the one that I'll discuss in a little bit of detail, which is moving now from the microscopic to the macroscopic, and now how can we use data science techniques at the level of the whole body, particularly in the area of surgery. So you might not know, but Johns Hopkins is famous as one of the best medical institutions in the world. And one of the things that happened at Hopkins was that one of the first surgical training programs was created back in 1889 by Sir William Halsted. And when this training program was created, the question of how do we teach surgery came about. And the main principle, and is written here in this slide, was the apprenticeship model, where you see one, do one, teach one. Mainly, you see how someone is practicing the art of surgery. You try it yourself, and then you, if you are able to do it, then you can teach someone else how to do it. The question is, 150 years later, does this approach scale? Are we able to teach and train the next generation of surgeons? Do we have enough for the growth of the population? And this is becoming increasingly difficult, particularly because even the time for teaching relative to the time for practicing, there is competing interest. And so, moreover, many things go wrong in many surgical procedures. You might not know, but from 2000 to 2005, there were more than 40 million procedures done every day. And on the order of 30,000 deaths are attributed to errors that happen during surgery. And even if the person doesn't die, there could be just dramatic increases in cost due to extra hospitalization and whatnot. So this question of how do we improve and accelerate training, and ideally with the goal of reducing and preventing some of the errors that can happen, is very important. So one approach to addressing this issue is the idea of can we use robots to do surgery? And if one uses a robot, the idea is that one can reduce the number of the recovery time. And that's primarily because, say, there is less tremor, there is a single incision point, you can allow the tools to pivot around the incision point, so the incision size is much smaller. The challenge there is that learning how to do surgery with the robot might take longer, and therefore further increasing the training time. So under the leadership of Greg Hager at Johns Hopkins University, a large collaboration began at what we called at the moment the language of surgery, which was the question of how do we take videos of surgeries as well as data from the Da Vinci robot in order to assess the level of skill of a surgeon at doing surgery. And that requires building a lot of machine learning models, and because you need to model expertise, you need to model what does it mean to do a surgery? Can I take a procedure, divide it in tasks, maybe look at the gestures of movement patterns that a surgeon is doing when he or she does surgery? And how do we model all that? And how do we come up with a score as to how well someone is doing during a surgical procedure? So I hope, and where in person I could do these tests in real time, but the question for you is, I'm showing you two videos of someone doing a suturing activity, who is the expert and who is a novice at doing the task. And I guess you will quickly appreciate that the person on the left sort of immediately inserted the needle in the dot, while the person on the right, it came out in the wrong place. It took maybe a little longer. Sometimes actually the time it takes is not the best indicator, but rather the precision and the smoothness of the movement. But anyhow, I think you as a human in this particular case are able to appreciate that you could potentially give a score and very easily determine that the person on the left is an expert and the person on the right is not. But how do we do that automatically was the question. So the idea was, and that's why the project is called the language of surgery, is can in the same way that we can model speech and divide what I'm saying into sentences and sentences into words and words into phonemes, and what I'm saying makes sense because there is a grammar that orchestrates how all these phonemes are put together and what word goes after each other. The same thing happens with surgery. You might have a 2-hour procedure. It's decomposed into a sequence of tasks that need to be done. And every task, in this particular case, suturing, is also decomposed into gestures, like grabbing the needle, inserting the needle, passing the needle from the right to the left. And once you do that, you need to pull the needle up and then passing the suture from the left to the right. So therefore, we needed to build something that would detect each one of these gestures or these basic phonemes. In fact, we call them sergenes with that analogy. And then based on how well you're executing these sergenes, can you provide a score like a number, say between 0 and 15, I think is one of the typical scores that is given to doctors of how well this is being done. And how could you assess skill? Well, I hope you appreciate that, and we actually saw that experts for suturing would just go from grabbing the needle to inserting the needle to getting the needle out on the left and pulling the suture. And so the probability of transitioning from one surgeon to another was very characteristic. While a novice would tend to make a mistake and the suture will not come out in the right place, and therefore they would go back. So these just transition probabilities between these hidden states that correspond to different surgeons would already be indicative of skill. So naturally, we began this project before the deep learning era. So this was around 2009, 2010. At the time, hidden Markov models were very common for this type of modeling. We extended this to using conditional random fields. And so a lot of spatial temporal probabilistic graphical models was what was being done at the moment. Later on, we moved into deep learning style approaches, particularly those that are developed for video and action recognition, where you begin, say, with the video of a surgical performance, you use a deep network to extract features from the video. And then you have a temporal neural network that models the temporal evolution of those features. That being said, though, it was not enough. And it was not enough because these deep networks are meant to operate at a certain temporal scale. And when I was talking about the question of skill, I said, oh, you need to transition from grabbing the needle to inserting the needle. Those probabilities of transition occur at certain scale. It may take you, say, a second or 2 seconds to insert the needle. And how long it takes you varies from video to video and from execution to execution. So the transition probabilities are between latent states, and we don't know the length of the segments. So this segmental component that you see at the very top is still a probabilistic graphical model that you put on top of a deep network for being able to do this higher level inference at the level of segments. And this is another architecture that we developed later that just processed the whole video into an encoder-decoder style architecture, where at the bottom, you've got the input video X, and you map it to a small number of latent states. Imagine actually, for simplicity, that you might have a video that might have thousands of frames, but there are only, say, 10 different gestures in that video. So maybe the latent state needs to be significantly smaller. And therefore, the decoder, what it does is to predict or replicate those labels in all frames so that you can get Y at the very top is for each frame what gesture is being executed. So just to give you a little bit of a sense of how these different models work. So the first row, the first group, just to tell you, is what if you do this only out of kinematic data? Kinematic data is data from the robot. So in particular, we're looking at position, velocity, and whether some sensor or the gripper is on and off. So that's about 78 variables. So that's what sensors means. Video, it's at the bottom. That's just looking at the video alone. Can you make predictions? And then the different models, the one that says CRF is for conditional random field. So that's a little bit classical pre-deep learning. Then the spatial temporal CNN is the integration of these conditional random fields with convolutional networks. And the final is the encoder-decoder architecture. And then there is two ways of measuring accuracy. The first one, accuracy itself. For every frame you measure, did I get the gesture right? Yes or no? And the edit distance is another that is really measuring at the level of segments. Did I predict the sequence in the right order correctly or not? And I think the main point is that we were able to achieve an accuracy on the order of roughly between 80 and 90%. And the methods based on these encoder-decoder architectures were the one that ended up performing the best on nearly 90% in terms of the edit distance. And similarly for videos, I hope you appreciate that performance is a little bit low on video alone versus the sensors alone. I don't have the numbers here, but we got the best performance above 90, 92 or 93 by putting together both kinematic data as well as video data. All right, let me spend the last 2 or 3 minutes talking just with one slide, I think, about some of the projects we're doing right now. So this is now a completely different realm. We're looking now at rehabilitation therapy, and we're working a lot with toddlers as well as children, and even in some projects with adolescents. In the case of toddlers here, they may have a motor disease. Think about cerebral palsy as one example. And what that means is that they have difficulty doing basic movements. They may not even be able to hold their own weight, and therefore that delays their ability to crawl, their ability to go upstairs or walk, go upstairs in an incline and things like that. So the current therapy for that is that they put them on this harness that you can see on the left. The reason for putting them on the harness is that the harness is holding the weight of the baby, and they don't have enough strength to stand up. And so with that harness, which is connected to a parallel robot, that allows them to move in the entire playground, in theory, without having to hold their own weight. And now they really play. And the idea of playing is that they attract them to something interesting that allows them to move from point A to point B. And the therapy is to get them to move, initially to just crawl and different levels of crawling, and eventually to crawl up in the inclined plane, and eventually to stand up and whatnot. So here we wanted to go even farther, even though we didn't get there yet, to put a robot in the play area that, and therefore ask the child ideally to imitate the movement of the robot. Right now, they're really imitating the, or being directed by the caregivers. So how do you assess that the child is making progress? Even today, in these therapy sessions, they put some cameras, and behind the scene, there is an operator that is looking at the cameras. And they have some software that allows them to manually click on the different limbs and manually mark what kind of movement was done, so that then they can get an assessment and a score on how much they were able to move. So the goal of this project is, because we're not there yet, to be able to assess the movements of the child directly by looking at the videos. And so that requires you to detect where the child is. That requires you to be able to estimate the limbs and then do motion analysis. And you might say, okay, well, just throw computer vision technology at it again and it's going to work. You might think, well, why not use Kinect? And so Kinect was trained and developed for adults for playing games, and it's not well trained for children who are going to be in very different poses, like crawling. There is not a lot of training examples on the web of babies that are crawling. I mean, it could be collected. Also, because of the environment, there is a lot of occlusions. Typically, you're going to have the caregiver and many other people. So developing a multi-camera system that always has one view that is unoccluded, it's something else that we exploited. Anyhow, I don't want to give more details. We're running out of time, but that's, I think, the idea, to use computer vision technology for rehabilitation. So to summarize, I think all I wanted to say is that I think there is a great opportunity to develop AI techniques for healthcare. We do have a lot of data, but the data is not annotated. We need to develop much better methods in terms of the ability to operate with limited labels and exploit domain knowledge in order to get AI to work in those domains. There is a lot to be done regarding making methods interpretable, regarding ensuring guarantees of accuracy, fairness, and so on and so forth. Let me conclude by just saying thank you. Thank you to you for coming. Thank you to my many colleagues at Johns Hopkins University. I have the pleasure of being in one of the best biomedical engineering departments. And pretty much everything I've done, it's only possible due to the fantastic colleagues that I have at Hopkins that work in many areas of medicine and that allow for these collaborations with data scientists. Thank you very much, Carol.
Thank you so much, Rene. It was a really inspiring talk, really bringing together applied mathematics with biological, medical expertise and machine learning. So it was really wonderful. Thank you. So I'm just going to remind everyone how to ask questions. So please either type your questions into the Q&A box. Either I'm going to read them, or if you type live at the end of the question, we're going to unmute you and you can ask your question yourself. So before I'm going to the Q&A box questions, Rene, I might start out with a few questions. So the first one is actually going back to the very initial project you've talked about, the hematology project, which I found super interesting. So in that, have you thought about using image transfer between the microscopy images and the lens-free imaging to maybe enhance the information in the lens-free imaging? So as a kind of offline thing that you do before to train a model to recognize correlations between the lens-free images and the microscopy images?
Yes, and we did do that. So we built in fact, in order to actually do a better assessment and evaluation that would compare not only with the final concentrations, which is a little bit of an indirect measure because you've got to split the blood into two streams, the one that you put into the hematology analyzer and the one that you put to the new imaging device. So you're never actually comparing apples to apples, right? And so to compare apples to apples, you need to build a hardware setup where you have regular optical imaging with the other, and you're able to image ideally the same image at the same time. Building the hardware is a little bit complex, but then even once it's built, you need to do some sort of matching of what cell corresponds to which one.
Yeah.
So we did all of that. And I don't remember whether we published it or not. And if we didn't, probably I shouldn't say too much because then it belongs to the company. So let me stop there.
Okay, that's super interesting. You could even maybe imagine that if you don't have paired, because that's what you're referring to, right? You don't necessarily have, it's difficult to get paired data between the microscopy and the lens-free imaging.
That's right.
Yeah, you could maybe even think about doing something that doesn't look at paired data, but maybe is happy by looking at distributions, the different shape of distributions that these two type of images produce. But yeah, but that's really, really interesting. Okay, clearly you thought about it. So then about the evaluating the surgeon. So there were two thoughts that came to my mind when you talked about this. So one was this could be also thought about a bit in a controversial way, because you are letting a machine evaluate a surgeon, and you could they're probably thinking about the fairness aspect that you have pointed to at the very beginning of your talk, the fairness of AI. I wonder what your thoughts are on that. And then the other thing that came to mind was somehow, what's your vision for this? Because if you think about the next steps leading from what you can already do to all the way to full robotic surgery, there could be somehow a path, right? Because once you can evaluate a surgeon, you could have a machine maybe online while they really do a surgery correct, maybe, or alert them that they shouldn't do something or should do something different. And then next step could be really robotic surgery. So I wonder what your thoughts are on that.
So fantastic question. So my first point is, well, so what are the alternatives? How are surgeons evaluated today? So by and large, and you'd be surprised, the main metric is that you need to practice by a minimum number of hours. So that's the first one. And I always joke in some of these talks by saying, well, if I was playing soccer, no matter how many hours I would practice, I will never get to the level of Messi, right? So the second metric is, of course, they're evaluated by having someone that is teaching them and providing them a score and giving them feedback, as with most apprentice style models. So is the goal really to replace and to give people automatic scores? I don't think that that would be necessarily the right approach. But compare the situation where maybe I have the robot and I practice on my own and no one is giving me feedback. And maybe my videos are recorded and then given to an expert who then looks at them at night and gives me a score. Versus a situation where I could potentially have some tools that immediately tell me, oh, you did this well, you didn't do this part well, and maybe show me a video of how I should have done it. So then maybe the final evaluation and the final score will always be given by a human, but the process of learning would potentially be accelerated by having these tools, even if they're actually never used for formally evaluating surgeons.
Yeah, okay, super interesting. Thank you, Rene. So I should have been probably more polite and start with the Q&A box questions, which I'm now going to do. So there is a question by Naeem Somro, who is asking, we are undertaking a trial in the UK to capture video data from real surgical operations and kinematic data across 6 surgical disciplines. Have you done assessment at the level to be able to make predictions on the outcomes and competencies? And linked up, but there is another question by Naeem, I'm also going to read that. Have you considered aligning electronic health record data with data from surgical robots to make more precise predictions on the surgical outcomes?
Yes. So regarding the first question, I wish I had Professor Greg Hager here because he would know the answer to the question. So I stopped working on this project around 2016, but he continued with the project. So at the time I was there, we still were at the level of, the data sets that I was mentioning were fairly small. They were maybe 50 trials. Maybe there were 5 experts, 5 novices and whatnot. Each one of them tried 8 times. At the time, Greg Hager was leading a data collection effort across multiple training sites in the US and across multiple procedures. And I know that he's continued on that line of work. And so if I had to predict the answer to the question is yes, this has been scaled to the level that one can determine not only the gestures at the level of a particular subtask, which is what I was talking about, but more broadly, looking at whole procedures that are for 2 hours. And also potentially correlating at which stage you are in the surgery with, say, a textual description of the task. How much is being correlated with actual outcomes and assessment? I don't know the answer, so I apologize that my answer is very incomplete.
Thank you. Thanks a lot, Rene. The other question is by Kerstin Eder, who asks, which techniques do you use to provide assurance of the safety and security of these safety critical machine learning based techniques, which goes back to, I guess, your interest into the foundations of machine learning as well.
So if I had to say the right answer is none. The reason being, these techniques are not currently deployed yet they're not being deployed. And so when it comes, for example, to the robotic surgery project, it's not that you have a robot that is automatically doing surgery. This is mainly for the purpose of training surgeons. And a lot of the training is done with avatars as opposed to with real patients. Now, how do you ensure the safety there? The AI techniques, as I said, they are help for practicing, but they are not being used as part of the procedure. And so therefore, That's why their safety is more, how do I design a Da Vinci robot that's going to do surgery safely, but that has no AI in it, right? And so, but the question is nonetheless critically important. How do we ensure that these procedures are safe? So we are working. I don't think we are at the level that these things are integrated and we really have a clear answer on how to do it. But there is a lot of research, and I have at least 2 or 3 projects on the robustness of deep networks to adversarial attacks. So we are working on the question of how do we build defenses? What are the weak spots? How do we have certificates of guarantees of performance with robustness to small perturbations and whatnot? But I don't think any of that is yet at the level that can actually be deployed into a real system. I mean, we see over and over how the performance can go from 90% classification accuracy to 0 with an epsilon modification to the input. I think there is a long way to get to the point that, one, we really understand how to measure safety, and 2, how to ensure that the predictions are safe. And even when we do, in theory, another thing is deploying the real system, right?
Yeah, I agree. I agree. There is another question from Naeem. So thanks, CSATS currently can segment surgical video data and provide Gear's score for each segment. That's, I guess, a comment. So there is another question by Lawrence. Are the main problems in the area of developing better learning algorithms in gathering learning data, in transforming the data, or in annotating the data items?
I think
they're all connected
both.
somehow, but yeah.
I think they're all connected. They're all connected because, I mean, in the blood project, I would often get the question, how many annotations do you need? What kind of data do I collect? And as data scientists, it's a little bit embarrassing because the current practice is really, well, you give me some data, it's already annotated, I train a model, I make a prediction, I compute the errors, and then based on the errors, I figure out I need more data for this or for that. And because building error bars on top of deep networks predictions is still under development, we don't have a sense of the number of samples that is needed. Then in medical problems, there is a lot of variability, right? The prediction for the blood tests, whether you brush your teeth or drank alcohol, those numbers change. And so the variability due to external factors is so large. So how do you collect data? You could have, and how do you collect data when you know that you're only going to be able to annotate a small fraction? So you could even begin thinking about developing methodology that you would do this in an iterative loop, where given where you are in your prediction errors, you would decide what you need to collect next, and which one should be annotated versus which one should not be annotated. I'm leading a MURI between the US and the UK, actually, called Semantic Information Pursuit. That is a little bit along these lines of analyzing data by having a sequence of queries. And the queries could be, in principle, not just, is there an object in this region of the image, which is what you do when you're trying to parse a scene, but rather, should I collect this data or this data? Should I annotate this or not? Or grabbing a label for this bit and piece of this modality. Getting to the level we can do that, so IBM Watson can do that with text only by having this sequence of questions and answers, but being able to do that with multiple data, I think, in my opinion, is the way to go to address this question of actively collecting and labeling and retraining models in a situation where we cannot live in the current paradigm, which is that I collect everything, Then I trained the machine learning models, and then I realized that they didn't work, and it was not enough.
Yeah, thank you very much. So there is another question by Muhassin Besoman, and thanks a lot for the nice talk. In the brain neuronal activity application, how do you collect the noisy images in the first place? Invasive procedure or MRI, CT scans? If invasive, what's a concrete application?
I don't know all of the details of the acquisition, so I could be wrong. I know the details are in the paper, but my understanding is that this is calcium imaging of mice, and it is invasive. So this is done on mice.
Mm-hmm. Yes. Okay. So that's on animal models. Okay. And a follow-up question from Lawrence: with the thumb prick method of taking blood, even having used hand cleansing liquid, which often contains alcohol, can contaminate the reading. That is very relevant during the pandemic. Is that a problem? Is that a problem for you?
So I don't know the answer to the question, really. That's not my expertise. But I think it may depend on what test you might be doing. That might be referring to whether you have something on the blood beyond the basic counts that we're talking here, which is red blood cells and white blood cells.
Yeah.
So if what the person asking the question is referring to is that might change the fact that there are other elements in the blood, and that those could be confused with some of them. So that's a valid point. If that changes the concentration, but I don't know enough, right, about the specifics. That needs to be a question of what is it contaminated with, what is the size, is it visible? There is a lot that goes into it. Yeah.
I mean, it could. I also don't know, but you could even imagine it changes maybe the contrast of or some properties of the image. I don't know. Another question by Hong Wang, also towards your hematology project. One image can contain hundreds to thousands of blood cells, which makes it almost impossible to annotate, as you actually also emphasized, I believe. In this situation, then, how to evaluate the accuracy of cell detection done by deep learning? So how do you compare with the ground truth?
So there are various ways of answering the question. The first one is the following, where at the end of the day, for the blood test, you really need to produce what is the concentration of red blood cells. So if the number of cells is very large, whether you detected each one of them perfectly or not is not that relevant, because you are producing an average number. And so in terms of the evaluation, the way we were doing it is exactly as I said. You take a blood sample, you split it into two. One of them goes to the hematology analyzer that will give you the concentrations. The other blood sample goes through the microfluidic device and is imaged, and we produce the numbers, and we do the comparison at the level of statistics without having to have a metric for evaluating detection per se. Now, you're totally right that there are challenges that pertain to the concentration. If the concentration is very high, the reconstruction algorithm could fail, and therefore the detection could fail. So ideally, you would like to be in this situation that is a little bit more sparse, that the cells are sufficiently separated. So to do that, you can actually add chemicals to the blood and do dilution. In fact, this is very relevant because also the concentration of red blood cells is dramatically different than that of white blood cells. It's like a factor of 1,000. So another challenge you face is that if you do dilution so that you can see the red blood cells, then the white blood cells disappear almost. If you dilute by 1,000, right? Then now you might have 1,000 red blood cells for one white blood cell. So all of these challenges need to be taken into account. And so that's what I always tell my machine learning students, that sometimes I think that throwing machine learning to problems would solve the problem, but there is real value to working with biologists, clinicians, and expert domains, because many of the solutions to making this work wouldn't have occurred to me or to us purely as machine learning person. Like this idea, well, you can do dilution, or you can do this, or you can do that. That's all the domain expertise.
Yeah, and if you do this cleaning, I guess, of the images to just represent one cell type, these represent somehow the ground truth, even if these images look a bit look different. Yeah. Okay. Okay. Super interesting. There are many more questions coming in, Rene. I hope this is okay. Another question by Namit Stillman. Really interesting talk. Would you be able to elaborate on the type of generative probabilistic model you use to classify cell types? Was this a deep generative model or a more traditional model like a Bayes classifier?
Yes, I don't have the slide in front of me, but at the time it was really a sparse convolutional model. It was not a deep generative model, in that maybe I could even just go back to the slides if I may.
Yeah.
Go back here. Yes, so here it is. Yes, so by and large, if you wish, it was simply, you can hypothesize that I of x, that you have a conditional distribution of P of the image and all of the other variables, and you just factorize the model is probability of the image, given that I know the classes, that I know the locations, and that I know the numbers, and so on and so forth. And for this image model, you're totally right. So first of all, for this, I think we use a Poisson. For this, I think we used a not a Bernoulli model, but something that models distribution between 0 and 1. But the key thing, the key place where we could have used a deep generative model would have been for the image model. But because it was sparse, we instead just use a Gaussian distribution of the intensities, where it was really a sum from 1 to the number. The mean, for instance, was the sum from 1 to the number of cells of the convolution of the template with a delta at the location of the images. So something extremely simple like that. And the reason we could do something extremely simple is because the reconstructions algorithm that we had produced was able to give us these not binary images, but images that have an intensity value between 0 and 1, that really was, the background was nearly 0 and the cells were nearly 1. So the need for having a more sophisticated deep model was not needed. But also at the time we were doing this, the deep models, well, we didn't have enough data to train these deep. That was another thing. Right? We were always restricted. How much data can you collect and to have? We did do, actually what we did do was to train a deep generative model for platelets, which we did using synthetic data. And we wanted to distinguish platelets from red blood cells we actually had a 3D model of how red blood cells look like. We were able to synthesize and generate a lot of synthetic data that way, that then we could use to train some of these models in those cases. But anyhow, I think the main point was we were trying to adjust the kinds of model to what can be learned from the data that we had available and from the labels that could be collected. And therefore, not all the battery of existing machine learning models was really applicable.
Yeah, okay, this makes sense. This makes sense. So a last question, Rene, from Kerstin Eder, again, as a kind of follow-up to the safety question. So the question, I guess, really is how important is it to really think about the safety issue already when developing the methodologies versus leaving the safety considerations to the very end?
I think that's very important. I think let me move into a very different domain, but the domain, say, of robots that you want to deploy or self-driving cars. I think in there it's very clear. You need to think about it from the very beginning. You need to look at what are the probabilities of errors of detecting pedestrians. How does that change with the weather conditions? Leaving that to the end, I think it's too late. Now, do we have a general way of approaching safety? Well, for each problem, we might actually have different definitions of what it means to be safe and what are the quantities we care about. So I don't know of a very general way to do it with AI that would apply to all cases. And I think we are at the level that we look at each application on its own. We need to define from the very beginning what are the safety guarantees that we need to ensure, augmenting the AI models with variables that have a semantic meaning, as we did here, and correlating those semantic variables with the safety needs for that problem. And there is, I mean, in various ways, this has existed for a long time, but with the arrival of deep networks is having importance again. You need to do deep learning with constraints. And the constraints can be constraints that are based on safety or fairness, and this naturally means that for each application, you need to define what safety means. And when you're training your machine learning algorithm, you need to ensure that those constraints are satisfied.
Yeah, so that's, I think, it also the impression that I got from your presentation, that bringing together deep learning with mathematical models as constraint that are inspired also by the biological expertise. I guess they're both for practical reasons, because you don't have so much annotated data and so on, but also for reasons of guarantees, of actually being able to deliver guarantees for these methodologies. So really interesting. Thank you so much, Rene. I think we are indeed at the end of our time. So I'm going to close the session now. Thank you very much, Rene. It was really very, very inspiring. And I think I'm echoing the feeling that the participants have also raised in their questions. So let me now wrap up. And thank you all for joining. Before we say our final goodbyes, don't forget to check at our website for upcoming lectures. The next Turing lecture will be given by Dr. Nicole Turner Lee on the 14th of July. And tomorrow we'll be joined by Kate Crawford in conversation with Judy Watchman to discuss her new book, Atlas of AI, at 12 o'clock. And next week, we'll be celebrating Alan Turing's life and legacy at Breaking the Code, a panel event including Alan Turing's nephew, Sir Dermot Turing, and TechMoms founder, Professor Sue Blake, OBE. You can register for all these events and others now on our website, the Turing AC UK website. The AI Council, supported by the Alan Turing Institute, is seeking the views of the AI ecosystem to tell us what you think is important for the national AI strategy. To take part and have your opinions heard, scan the QR code on the screen now, please. And finally, on behalf of everyone at the Alan Turing Institute, I really would like to thank you very, very much, Rene, for joining us today. It was a pleasure to host you and to hear about your really inspiring and incredible work. Thank you.
My pleasure was mine. Thank you so much, Carola.
Bye, Rene. Thank you. I think we should leave. Bye.
Bye-bye.